Okay, our focus today is fluorescence microscopy and Parkinson's disease. And I'm Cami.
I'm Mackenzie.
And I'm Laisha.
Okay, so ever since the first published academic paper on Parkinson's disease in 1817 by Dr. James Parkinson, scientists have been furiously researching how this neurodegenerative disease takes over the brain and the motor functions of a human affected by it. As scientists continue to struggle with finding the cause of this disease, they began to study the changes the body of an infected person goes through and discovered that the brain severely decreases its release of dopamine after an infected patient reaches around 60 to 65 years old. Despite knowing the symptoms of the disease, the scientists have not been able to figure out why the sudden decrease of dopamine in the brain occurs. Recently, scientists began studying the brain using microscopes in order to search for ways to cure this disease or for a stronger and more effective medication to lessen the symptoms.
Okay, so our focus question is, how can fluorescence microscopy help advance current research in Parkinson's disease? And before we go into the answer to that question, first I think it's important to talk about fluorescence microscopy. So it's a general term for a really complex concept, but it's involved in a number of techniques that benefit the scientific community. But the principle of it is applying colored dye to subunits in order to help distinguish their locus or their function, which is a really important tool in comparison. So the process works by adding a source of light in order to excite electrons to a higher energy state, and the level of which dictates the color that they show. And fluorescence, by this principle, is defined as the emission of light by a light-induced singlet, and that singlet would be electrons in this situation. And so Parkinson's disease is a neurodegenerative brain disorder, and the problem is that we don't know the root cause of it, but what we do know is that damage in dopamine-producing cells leads to the levels of dopamine dropping, which ultimately results in the symptoms of this disease. And there is some evidence today that shows that it is a genetic disorder, although it is not solid.
Okay, so the social impacts of Parkinson's disease. In the United States today, there are approximately 1 million people affected with Parkinson's disease. It involves the malfunction and death of vital nerve cells in the brain, and therefore symptoms worsen over time as the patient gets older. Some of the most common symptoms includes tremor of the hands, the arms, the legs, the face, and the jaw, slowness of motion, stiffness of limbs, and impaired balance and coordination. When nerve damage gets too severe, surgery may be necessary. However, medication is typically used to help lessen symptoms. Although scientists are unsure of what causes Parkinson's, some speculate that it is related to clump proteins in the brain. And these proteins, there are two of them that we kind of talk about. It's alpha-synuclein and proteins from the gene LRRK2. If we can isolate these genes and image them over time, we may be able to create a drug that helps suppress the translation of the genes that encodes for these proteins and prolong the lives of the people with Parkinson's disease.
Okay, so some background and precedent on the issue, or on fluorescence microscopy. It was first defined by Sir George G. Stokes, and he coined the term in the year 1852. But further research throughout the 19th century confirmed his belief that organisms will fluoresce when put under ultraviolet light. And another important principle about fluorescence microscopy, and what may even perhaps be considered a limitation, is Stokes' law, which states that as the electrons come down from their excited state into a more relaxed state, energy is lost, and therefore it must be compensated by a shift in the light emission spectrum, which results in longer wavelengths. So we can compensate for this by using fluorescence filler combinations, which will kind of help us separate between excitation, which emits light. So our project proposal to sort of delve into the issue is working with the LRRK2, which is a gene that encodes for a protein found frequently throughout the brain. And it has been seen in the past to be linked to Parkinson's. So in past cases, they found that mutations of this gene will result in the damage in dopamine-producing cells. So our idea is to use a technique which is called fluorescent in situ hybridization, which stems from the concept of fluorescence microscopy. And it combines principles from both light microscopy and molecular biology by first targeting the DNA sequence of interest, which is the LRRK2 in our situation, and to then track which parts of the brain is found, use the fluorescence to stain these parts, and then we can see their location and make some important comparisons, which will hopefully guide our investigation into the cause.
So the Brain Initiative started in 2013. It was brought up by our former President Barack Obama, and what basically scientists wanted to do was look at more into diseases that were from the brain, which is like Alzheimer's, Parkinson, and other diseases. But some of the obstacles scientists have faced are that the brain is way too complex for them to look at, and it is too big for them to look at it completely, like altogether. And it is also the technology they use is not far advanced for them to look at the brain, and because they can't look at a brain of a person who is alive, it is more difficult to them to find what they need. So in order to advance research, what we're gonna do is we will color dye highlight particles. We'll highlight particles to see their location, and this we will use fluorescence microscopy.
Okay, so some important discussion questions surrounding the topic. One is, in the case that Parkinson's disease is found in the subject's genetics, what are some ethical concerns that can arise developing solutions for prevention or cure of this?
A second question that we have is, how would you be able to determine which protein is causing the decrease in dopamine, if it is in fact a protein? Because scientists aren't really sure right now what protein it is, or if it actually is the protein that's causing this. And third question is, who funds projects like this, and what are their motivations for doing so? And also, how much money should go into this research? Contributions. Yeah.
Questions? Yes. So how does Parkinson's develop throughout time in humans?
Okay, so basically what starts off when people, it's different for everyone, but if someone is infected with Parkinson's disease, it normally becomes prevalent around the age 60 to 65, and it starts normally off with the shaking of the hands, and then it keeps progressing to other shaking, stiffness of the body, just lack of coordination, and eventually, as the vital nerves in the brain keep dying because they don't get the dopamine, some people require surgery. There are medications that are used to suppress these symptoms. However, sometimes they don't work. It's different for everyone. And so if they do need surgery, it's called deep brain stimulation surgery. And basically what it does is it blocks electrical signals from target areas of the brain. So it kind of focuses, it kind of it just basically it doesn't harm healthy brain tissue by destroying the damaged one. So they have to do that. Yeah, so basically as they get older and older, they start to lose more and more function. It could also lead to neuropsychiatric disturbances, which could be disorders of speech, cognition, mood, behavior, and thought. So yeah, it kind of progresses over time to it keeps getting worse and worse as more vital nerve cells in the brain die.
I know we don't know the root cause of it, but is there any information on what damages the dopamine-producing cells, or what could possibly cause damage to them?
Sure. So I think what I think the problem that they find is that the cells are no longer producing dopamine. I think that
There's a photo. So if you look at the top, that's the normal neuron and neurotransmitter, like the amount of dopamine, and in the one below it, that's after, like when they're affected by Parkinson's disease, that is the change, I guess. But yeah.
Yeah, I think they don't know what's causing damage. They just know that they're not producing anymore, so it's probably somewhere along the tract where cells are emitting proteins. If that makes sense?
You mentioned at that point it's like the clumping of cells, or what? You mentioned something about clumping.
It's the clumping of certain proteins. That's what scientists today are currently looking into to see if that's the root cause of this lack or this decrease in dopamine production. Like, they're still trying to figure it out, but there are various proteins or clumps of proteins that show up in people who have Parkinson's disease, and so they're like trying to see, is this the root cause of it?
Where's it clumping at the nerve ending?
I think it's just in the brain. I'm not 100% sure where it's clumping.
Yes. Speaking to your challenges questions, one of them was, what are the ethical considerations in developing a cure? And I was curious as to what those might be now, because I remember probably before you were born when Michael J. Fox was diagnosed with Parkinson's, everyone was talking about the ethics of stem cell research, which is now part of the discussion here today. It's become part of the process. And so I'm wondering what else is currently at stake.
Okay, yeah. So I think there's the problem that other groups discussed, that testing on humans is really risky and dangerous. But then a whole separate entity is kind of altering DNA, because if we find that Parkinson's is caused by sequences in DNA, then that does give us the power to go back in perhaps an embryo and alter the DNA sequence in order to prevent Parkinson's from occurring in the lifetime of the child. But some people have religious concerns with this, thinking that it is unethical to alter DNA, and even though we may have the ability to do it, it could malfunction, and it might be very risky or potential to harm the zygote's life.
So you said that the proteins basically come together, and then for whatever reason, the dopamine becomes non-existent, or it's no longer there. If you could increase the amount of dopamine just by a pill, or there's pills now, do the clumps of protein not allow the body to store the dopamine? So if you could just increase dopamine, would that do anything good, or would the body still just reject the increased dopamine?
Yeah.
The protein, because of the clumps of protein.
Yeah. It really honestly depends on each person, because I know I read that the medication does really help some patients who have Parkinson's, because it does increase their dopamine levels, maybe not back to how it normally is, but get it back close to that. However, it does sometimes not work. I'm not 100% sure if it's the proteins or not, because they're not 100% sure that's the reason why dopamine levels dropped. However, I do know that does help with some, and with others it doesn't. And it really just depends on the patient, how they respond to the medication.
So with identifying the locations and regions of highest interest, do you think that would be able to help you identify later what is wrong with those particular cells by zooming into them? In other words, finding the right cells that are the cause of the disease versus all the other cells that aren't really participating in it?
Yeah, I think that could definitely help especially because we know that there are a lot of different types of cells in the brain, not just like in function, but in purpose. Or, I mean, those are kind of the same thing, not just in location, but in function. So I think once we track the kind of cells that are not emitting the protein or that are maybe not doing something they're supposed to do, it'll help us like track Parkinson's development, but also help us kind of with the brain mapping as a whole.
I just got a question. Now, Parkinson, it's like the neuron die, or they're like misfiring, or they're not communicating with each other. Is it like bad communication, or is it death of a neuron?
It's like a neuron's failure to do its job or like produce a certain protein that it's supposed to be producing.
Okay, because I mean, there was this other group that was talking about the flow of neurons or whatever. I mean, do you guys work with them and develop new ways of healthier ones communicate with each other or something like that? I mean, I don't know. That's just my
Yes, I think that's also really difficult to do because then you'd actually have to go into the brain, and that is also like one of the things when you do have surgery, you're trying to like redirect away from the damaged nerve cells to the like healthy ones, and so I think that's part of it. But it's really kind of hard to tell without like going in and like monitoring the brain for for a certain amount of time.